All right, so this is lab unit number 4. Focus here for this lab manual, or this lab unit, is going to be the brain, both what would be illustrative pictures of the brain as well as some pictures of a dissection, cranial nerves of the brain, and then we'll also do the eye and the ear. We'll have illustrative pictures of the eye and the ear as well as pictures of an eye dissection. So those are all the things, sort of like several little parts to this last lab. And I just want to run through it. I think by now you guys are pretty much know how these things go and what my expectation is, but I want to go ahead and run through it. So this first slide is just, some background on the brain and the cranial nerves, and basically what you're going to want to do is, of course, identify all the things in the boxes as we've done typically. First are just the regions, cerebellum, cerebrum, brainstem, different parts of those, as you can see listed in this picture here. Then the different lobes of the cerebrum, frontal, parietal, temporal, occipital lobe, which you can see clearly in this picture is sort of color-coded. Then on to the different fissures and sulci. The lecture portion covered a lot of this stuff, but basically fissures are deeper grooves that are separating the cerebral hemispheres. So like a longitudinal fissure is going to separate the left and right hemispheres. Transverse fissures is going to separate the cerebrum from the cerebellum. So transverse fissure, for instance, would be down here. Sulci are just shallower grooves. The central and lateral sulcus are the two primary ones that I have listed there that you want to make sure that you're aware of. And those are important because in front of the central sulcus, as you're going to see, we have different areas of the brain, for instance, like the precentral and postcentral gyrus, in front of and behind the central sulcus. Now, again, we go over the significance of that more in lecture, but you want to know that as well here. The gyri, as I was just mentioning, the elevated ridges on the whole surface of the cerebrum, all those folds are the gyri. And as I said, the precentral and postcentral gyrus are just the portions in front of the central sulcus and behind the central sulcus. In front of the central sulcus, the precentral gyrus is the primary motor cortex. In other words, where you initiate skeletal muscle contractions are initiated in this part of the brain that travel down the brainstem through the spinal cord out to the muscle. And the postcentral gyrus is the primary sensory cortex. Again, we go over this in the brain chapter, but the postcentral gyrus, of course, is where primary sensory information is going to sort of be sent to and relayed and interpreted, etc. Cerebral cortex is the superficial gray matter on the surface of the brain, as you can see in this picture. This have sort of a smaller picture there located in the cerebral cortex. We'll also see the cerebral cortex in the brain dissection. When you do a frontal section, it looks just like this picture here. Do a frontal section of the brain on a dissection, you can see the cerebral cortex as well, that superficial gray matter. All right, so on to the next, white matter inside the brain. Can't see it as well from this picture. The dissections will show it a little bit clearer. Corpus callosum is a big band of white matter that connects and links the two cerebral hemispheres. Fornix, anterior commissure, arbor vitae are the branches of white matter that call arbor vitae means tree of life. And you can see the arbor vitae here inside the cerebellum, all the little white branches there. So that's a pretty distinctive feature of the cerebellum. So we make sure you have all those tracks of white matter. And then parts of the diencephalon, which I think are all pretty well labeled, like the thalamus, hypothalamus, mammillary body, pituitary gland on this picture would essentially be right about here. Can't see it quite on this picture, but one of the dissected pictures, you can see the pituitary gland. And then the infundibulum is a stalk that links the pituitary gland with the hypothalamus. We're going to learn more about the pituitary gland and the hypothalamus and that relationship in the endocrine chapter that you'll be studying in this last unit as well. Corpora quadrigemina, again, we went over that in lecture. It was a big part, not a big part, but a significant part of the midbrain or mesencephalon that we talked about, and that's basically right here. Has two essentially bumps, a superior and inferior portion. We call them the superior and inferior colliculi, and those portions regulate sort of reflex responses from our head, neck, and shoulders to either a visual or auditory stimulus, as we went over in lecture. Then the ventricles, ventricular system, again, as we went over in lecture, are basically the internal chambers within the spinal cord, and they travel up the length of the spinal cord through the brainstem, and they expand in these larger chambers called the ventricles in the brain, and they contain fluid, as you know, which is cerebral spinal fluid. And so what you're wanting to identify here are the different ventricles within the brain. And again, remember, it's all continuous, starting up at the top with the lateral ventricle, traveling through the third ventricle, fourth ventricle, and then ultimately down to the central canal, the spinal cord. So it's all linked together. So I think this picture shows it pretty well. You can also see some of the ventricles up here in this picture. All right, so that's it for sort of the illustrative stuff of the brain. And then we're going to next look at the brain dissection. So it's unfortunate we can't do a brain dissection. It's one of the coolest things we do. In the semester when we actually get some tissue and actually separate it and appreciate a lot of the different features. So we'll just have to do it with images. And essentially what you're going to do, or what you would have done, I should say, in lab, is you're going to appreciate a lot of superficial features of the brain, and then you would basically have done two cuts. You would have done a sagittal section, which basically divides the brain in half, and then you also would get a second brain, and you would do a frontal section like this, and I'll show those different sections in a minute. But basically you would do two brains and have it that way. But so the first thing you want to do is appreciate the surface features, and the first thing you would do, your brain would actually come wrapped in the meningeal system. So you'd have the outer superficial dura mater, which, as you know, is the thickest outermost covering of the meningeal system, and you can see it pretty clearly on the image in your lab, which you can see. It almost looks like this parchment paper or something. It's completely, they didn't cut it off of this location. Typically, when you try to remove the meninges from that lower portion, basically like the hypothalamus and the pituitary gland and the optic chiasm and the optic nerve and all those things just sort of get peeled off with it. So oftentimes it's best just to leave it intact, which is why you see that. And so basically I've included a ventral surface image of a sheep brain dissection and labeled a lot of the things, and you can see them listed here, and you can just identify the different things as you go through that on the dissection. Then you would move on to the sagittal section. As I said, you cut it right down the middle basically, and you open it up, and on the inside you're going to see lots of different features. A lot of the things that you went over in the illustrations, or that I went over in the illustrations, you can see here. Like, for instance, the arbor vitae, you can see pretty clearly over here in the cerebellum. You can see a ventricle right in here. That's the lateral ventricle. You can see the optic nerve, ultimately would be the optic chiasm there. Here's the pituitary gland really nicely. So anyway, they're all labeled there for you. Just roll through them, identifying all these different features and structures, and I'll use these images on the exam. And then it continues on all the way through 23. So 1 through 23, this picture is pretty clearly labeled, and you should be in good shape. And again, a lot of it overlaps with the illustrative. Like in lab, what we typically would do is do models, and then you also do a brain dissection, so you can sort of compare both. Then we'd move on to the frontal section. The second brain, you do a separate frontal section, that I showed you in the previously. And as I said, you're just going to identify a few things, as I've had listed here, 1 through 8, like the cerebral cortex or the superficial gray matter, you can see pretty nicely. Let's see, this is probably like a good picture of it right here, right? All of that gray matter at the surface is the cerebral cortex. White matter, of course, is scattered about deeper. Longitudinal fissure, of course, separates the two hemispheres. We talked about that. The corpus callosum, you can see, is a band of white matter that links the two hemispheres. You can see the thalamus, lateral ventricle, and you would be able to actually remove some choroid plexus from the lateral ventricle, but it's kind of hard to see on this picture. So that's one of the things you would kind of lose out on, unfortunately, with the not being able to dissect it. All right, and so that's it on the dissection. Just make sure you're good on all those images and you'll be good on the dissected stuff. Then we move on to the cranial nerves. I'll use this image to have you identify the 12 cranial nerves, as you can see on this picture. So you definitely want to know this picture, and then you need to be able to physically identify them, but then also give me one function. So there are 12 cranial nerves, 1 through 12, and some of them will have multiple functions, but all you need to do is give me one function of these cranial nerves. So for instance, olfactory is the first cranial nerve. So you want to be able to identify that on that picture, and then I could ask you, what is a function of this cranial nerve? And you would write, you could write smell and keep it that simple. So just need to be able to do that in terms of the function. The type is really just more to give you a feel for whether it's sensory or motor type of cranial nerve. So essentially you need to be able to identify all 12 by name and visually, and then give me a function of each. All right, so they go through the rest of this lab manual. I think the picture here, it's kind of broken up, but hopefully in your lab manual, it's a little bit easier to read as you go through these. 9, 10, 11, and 12. And then I've included this mnemonic if you're interested in using that to help you memorize them, but you don't have to. That's what that is at the bottom. So anyway, so those are the 12 cranial nerves. And that is it for the brain. So you have the brain pictures or illustrated pictures, you have the brain dissection stuff, and you have the cranial nerves. Then on to the eye and the ear, the anatomy of the eye and the ear. I will use these pictures. As you can see here, the first thing you want to identify, of course, are the accessory structures like the eyelids and the angles of the eyes and the eyelashes and the lacrimal apparatus, which includes the lacrimal gland and ducts and canals, etc. So make sure you have all those. There are 6 extrinsic muscles that you have to identify, and one of these 4 pictures, you'll be able to identify all of them. Probably this one, this one, and this one, but that gives you a good idea of where those muscles are. And then you want to learn the 3 tunics. Again, we go over this in detail in lecture in terms of identifying the different layers of the eye, but you also want to be able to identify the anatomy portion of it. So fibrous, vascular, and neural or retinal layers that make up the eyeball. And you just want to be able to identify those. I'd probably use this as a great image right here to identify those structures. This is a nice image of the chambers for the anterior and posterior cavity. And then we'll move on to an eye dissection. This is a great experience you'd have to be able to identify the different parts of that you're not going to have, I should say, to be able to identify the different features of the cow eye dissection. But these pictures, I think, do a pretty good job of it. So here you just want to be able to identify the sclera and the cornea. The sclera is the white of the eye, of course, and the cornea is that sort of cloudy surface appearance that obviously where light travels through to allow us to see. So, all right, so those are the 2 things you want to see superficially. And then you have different structures like the vitreous humor or gel. Again, you can kind of see it here, sort of this gel that's found in the posterior chamber of the eye that helps support the shape of the eye and support the retina. The lens you can see pretty straightforward. The retina, of course, the choroid, tapetum lucidum. I think all those things are pretty clearly labeled, so use the picture for that. So I will use one of these 2 pictures or both of them potentially for the eye dissection. And then finally, the last thing is the ear, just identifying the different structures on the external, middle, and inner ear. And you can see they're all pretty clearly labeled in this slide. We have really nice models where you can see all these structures. Almost looks exactly like this image here. And so, as you can see, the external ear is out here, middle, and then internal ear structures. So just make sure you can identify all those things. And again, we go over those in pretty good detail with the lectures in terms of identifying how sound happens and the sort of physical process of distorting the auditory ossicles to stimulate the vestibulocochlear nerve. So you want to make sure you review those things as well. All right, so that is it. So this is Lab Unit 4, and happy studying. We'll see you.